Although bacteria and archaea are both characterized by the absence of a nucleus and are those classified as prokaryotes, it's important to know that analysis of ribosomal RNA sequences has shown that bacteria and archaea are almost as different from each other as humans are different from bacteria. So, for this reason, it's more correct to say that all organisms belong to one of three domains: bacteria, which is also known as eubacteria, archaea, which is also known as archaeobacteria, and eukarya. So this is more correct than classifying organisms into prokaryotes and eukaryotes. However, we will occasionally use the terms prokaryote and eukaryote for simplicity, and we'll clarify whether we're referring to bacteria or archaeobacteria if necessary. The bacteria or eubacteria include most of the prokaryotes we're familiar with, while the archaeobacteria include organisms that are adapted to living in extreme environments. An example of that are the thermoacidophiles, which live in acidic hot springs, and the halophiles, which live in very salty environments. Although the word archaea means ancient, that word is actually a misnomer. It's not a very good word for these group of organisms because evidence suggests, as you can see in this diagram, that archaea descended from a common ancestor that gave rise to eukaryotes long after diverging from bacteria. Archaea share many characteristics with bacteria, but they also have unique features as well as features that resemble eukaryotes. As you learned in general biology, one of the major differences between prokaryotes and eukaryotes is the presence of a membrane-bound nucleus in the eukaryotes and its absence in the prokaryotes. And that's the basis for the names eukaryote and prokaryote, with eu meaning true and pro meaning before, and the word karyon meaning nucleus. Right, so you already know that eukaryotes have a true membrane-bound nucleus, and prokaryotes have their DNA in the cytoplasm but folded into a compact structure that's known as the nucleoid. Now, keep in mind that the presence or absence of a nucleus is becoming a less important distinction than other aspects of cellular structure and function. As you know, eukaryotic cells have internal membrane-bound organelles like mitochondria, ER, and Golgi, and these organelles carry out different functions. Now, the membranes of these organelles have distinctive chemical compositions, so they have different lipids, and they contain different proteins that are needed to carry out different functions. Although bacterial cells have not been recognized to have membrane-bound organelles, internal membrane networks are present in some bacteria, like the cyanobacterium pictured here. In the case of the cyanobacteria, these membranes house the machinery needed to carry out photosynthesis. In addition to that, there are some bacteria that have organelle-like structures that can be either membrane-bound or protein-lined. And in fact, recent papers have described the presence of quote-unquote bacterial organelles like the magnetosomes that are shown in these images. These magnetosomes concentrate magnetic particles that allow bacteria to navigate in a vertical direction following Earth's magnetic field, and that allows them to reach low oxygen environments. Other studies have also reported the presence of interconnected membrane networks in some bacterial species. A distinguishing feature of eukaryotes, which has not been described in prokaryotes, is their ability to use membrane-bound compartments to exchange material both within compartments inside of the cell, but also with the external environment. Now, they do this via the processes of endocytosis and exocytosis, and that pertains to the exchange of material with the environment. In endocytosis, which is not shown here and which we'll discuss later in the semester, cells take up material from their environment when portions of the plasma membrane pinch off inside of the cell to form membrane-bound vesicles. Endocytosis is also used to remove receptors from the plasma membrane. The reverse process, where material is sent outside of the cell when vesicles fuse with the plasma membrane and release the material to the outside of the cell, is known as exocytosis. Now, in addition to these processes, eukaryotic cells also make use of internal vesicles to transport material along cytoskeletal fibers inside of the cell. Now, obviously, bacteria have to be able to exchange material with their environment, but they do this through the use of transport proteins in their plasma membrane, which are also present in eukaryotes. Another difference between eubacteria, archaeobacteria, and eukaryotes is the way they package their DNA. Now, the amount of DNA in cells is typically longer than the cell itself, so the DNA has to be packed into a small space. And that's true of bacteria, archaea, and eukaryotes, but eukaryotes have approximately 1,000 times more DNA than bacteria packed into the nucleus. And eukaryotes solve that problem by having multiple linear chromosomes. Now, although the cell does not keep the DNA so tightly compacted as what you see here throughout the entire cell cycle, we know that the DNA is segregated into daughter cells as compact chromosomes during mitosis and meiosis. Now, these multiple linear chromosomes in eukaryotes are complex with histone proteins, and as you may know, those complexes of histone and DNA are known as chromatin. Both eubacteria and archaeobacteria have a single circular chromosome, and they have some protein associated with those chromosomes. In the case of eubacteria, they have just a small amount of protein. In the case of archaeobacteria, there is a moderate amount of protein associated with their DNA. Another key difference between eukaryotes and the archaea and bacteria is the way in which the DNA is allocated to daughter cells during cell division. Bacteria and archaea divide via a process known as binary fission that is not very well understood. But as you can see, it involves the replication of their single circular chromosome and the allocation of equal amounts of that DNA and cytoplasm to daughter cells. In eukaryotes, following DNA replication, the DNA is compacted into chromosomes and is distributed equally among daughter cells through the process of mitosis, followed by cytokinesis. Now, it's important to point out that the cytoplasm is not always divided equally between daughter cells. So you could have asymmetric cell division in which one daughter cell differs significantly from the other, either in size or sometimes even in fate. Finally, the mechanisms of gene expression also tend to differ among these three different groups. In eukaryotes, the DNA is transcribed to mRNA in the nucleus, where the introns are removed, and that mRNA is then transported to the cytoplasm, where it's translated into protein. An important thing to know is that in eukaryotes, one mRNA molecule usually codes for one polypeptide. So although you see many ribosomes bound to this mRNA, they're all making copies of the same polypeptide. On the other hand, in prokaryotes, one mRNA will typically contain information for several different polypeptides. Also, because bacteria and archaea lack a membrane-bound nucleus, transcription and translation can occur in the cytoplasm at the same time. So you could have simultaneous transcription and translation because as the DNA is being transcribed into mRNA, several ribosomes can bind to it and begin translating it into protein. Now, in spite of these differences, it's important to note that the actual mechanisms via which transcription and translation happen in archaea are more similar to those that are seen in eukaryotes than in bacteria. I want to end this section of the lecture by pointing you to this table, which summarizes the key differences among the cells of the three different domains of life.